Hello, and welcome to this instruction course on botulinum toxin. I am Professor Mohammed Moin and working as the head of the Department of Ophthalmology at Postgraduate Medical Institute, Amiruddin Medical College, Lahore General Hospital, Lahore, Pakistan. The objectives of this course are that the participants should be able to describe the mechanism of action of botulinum toxin, identify the facial muscles for injection, make various dilutions, calculate the dosage of botulinum toxin, identify the injection sites on the patient, describe the etiology and treatment of hemifacial and blepharospasm, and enumerate the complications of botulinum toxin. Some facts about botulinum toxin is that it is the most poisonous substance known to man. It can actually be used as a deadly weapon in biological warfare, and 1 gram of crystalline toxin evenly dispersed and inhaled would kill more than 1 million people. Botulinum toxin is produced by the bacterium Clostridium botulinum, an anaerobic spore-forming strain commonly found in soil. And botulism is a form of food poisoning from undercooked foods that are contaminated with a toxin, most often found in ill-prepared and canned food. What is the mechanism of action? It irreversibly prevents acetylcholine from being exocytosed into the synaptic cleft, and the death of this part of the neuron may take a month or longer, although synaptic transmission ceases almost immediately. So the neurotoxin has got a light and a heavy chain. Here you can see that this is the molecule and it's got two parts. And this heavy chain is the part which actually binds with the cell membrane, causing it to be internalized as a vesicle inside the nerve ending. Then the light chain is released, and this cleaves specific snare proteins to prevent the release of acetylcholine. Here you can see the normal sequence of events in a nerve ending. You've got the synaptic vesicles which are filled with acetylcholine, and these, when they near the membrane of the nerve ending, they bind to the synaptic fusion complex, which is formed of snare proteins composed of synaptobrevin, SNAP25, and syntaxin. And as soon as they bind to this fusion complex, the vesicle is released into the neuromuscular ending, and the acetylcholine is available for action. With exposure to botulinum toxin, here you can see it is going inside and the heavy chain remains inside the vesicle. The light chain is released and the light chain is binding to the snare complex, thereby preventing the acetylcholine vesicles from being released outside into the neuromuscular junction. There are various types of botulinum toxin, type A, B, C, D, E, G, F. And the ones which we commonly use is botulinum A. So here you can see the botulinum toxin is present with a light and heavy chain, and the vesicles are prevented from being released into the neuromuscular junction so that acetylcholine is not available. And with time, actually, new nerve endings sprout so that the action of botulinum wears off. So why is it necessary for multiple injection of botulinum toxin? The Schwann cells for the damaged neurons secrete certain neurotrophic factors that keep the cell body alive and that stimulate regrowth of the axon. About 10% of the population is able to generate antibodies to botulinum toxin, and this only happens to people who have higher dosages and higher frequencies of injection. So it is recommended that you do not use botulinum toxin for intervals less than 3 months, and the dose should be kept at the recommended levels by the FDA. And the regrowth of axons can be likened to a regrowth of a starfish appendages, in that it is possible if the cell body is intact. And how does the action wear off? By the formation of snare proteins, decreased acetylcholinesterase, and upregulation of acetylcholine receptors. So in conclusion, the understanding of the potential of this sweet poison is very essential so that we can use it to our advantage in controlling spasms in patients with hemifacial and blepharospasm.